Hi, everybody. So I'm Matt. I'm from the I3S University of Porto. Um, and I wanted to talk to you today about something that's been rattling around my brain. May be a crazy idea, might be something okay. But I thought if I could talk to some people who know a little bit more about this would be a good opportunity, and maybe we can come up to something interesting here, right? Or maybe I'll find out this is insane and I should do something better with my life. Okay, anyway, so what I'm talking about, wanted to talk about was the possible role of pleasure at the origin of life on Earth. Okay, so we talked about a little bit yesterday. We talked about Gibbs free energy, right? We have pressure, temperature, free energy, pressure, volume, temperature, and entropy, right? And also enthalpy and entropy, right? So let's let's take a step back and think about what we mean. We're talking about the origin of life, right? And so there's a couple of basic sort of scenarios that are get tossed around a lot for the origin of life. There's the very common, the original theory once they discovered archaea was the idea of things arising in thermal pools where there's temperature because archaea were thought to be the oldest organisms. But then other issues came up with that very quickly. And so the possibility was, well, maybe if we have a thermal pool with a little bit of ice there, we can have melting of the ice and temperature gradients. So we can allow formation of more complex molecules at those interfaces, and we get interactions with clays. Um, and so whether or not the temperature had to be high or we could do it at low temperature and that was better was interesting. Um, Not entirely clear which is which, but then of course we also have the idea of black smokers down deep in the ocean, where you can get your thermal gradients, your chemical gradients, and even maybe a little bit of like small spaces in the porous part of the rock, where you can form sort of pseudo cells. Okay, right. And so there's a lot of basic ideas, but what's the difference between the black smokers and the thermal vents, right? The black smokers are deep in the ocean, the thermal vents are at the surface, and so there's a big difference in pressure. And now we've got this nice little four space diagram. And so we think about the fourth corner here, right? And we're really looking at two axes here, right? With the axis of temperature, of course, hot and cold, but we're also thinking about pressure as well, right? And pressure can also drive reactions, right? Because it's doing something very similar to what temperature is, right? And in biology, what really matters is if we're altering the activity of water. Okay, so and let's talk about what advantages we have here with pressure, right? One of the disadvantages of the black smokers is currently there aren't very many of them. They're in very small areas, right? So if we have these black smokers, and that doesn't mean that that's important, right? Because you only need life to originate once. But if you have a small area, there's less space for experimentation. Now, if we think about pressure itself as sort of the driving space here, we can think about the entire ocean floor. Okay, and so I wanted to talk about this, XKCD cartoon because I think it's a very interesting one. It gives us an idea. It's a good example of how big the oceans really are and how much depths we're dealing with here, right? So we talk about, and this for a vertical scale, this is very good. We can see, sperm whale hunting depth is about 3,000 meters. But what we forget about a lot of times is that the average depth of the ocean is about 3,800 meters, which is roughly the same depth that the Titanic is currently at, right? And so we know that that's very deep and under a lot of pressure. We saw that over the summer recently with the accident for the expedition there. Okay, but that's not even the deepest part of the ocean. There are deeper places with higher pressure amounts, right? And so we have a lot of experimental space to run the origin of life here, especially in the past, because the archaeal oceans were much bigger than the current oceans, right? Because not a lot of the water had managed to seep back into the crust. Okay? So let's go back to our history, what we're thinking about here, right? So we have the timeline of Earth, the Hadean, the Archean, the Proterozoic, Phanerozoic, right? We can roughly put time points on where things are, right? We know where the late heavy bombardment occurred. We can spot the difference between the Archean and the Proterozoic by the Great Oxygenation Event. Right? That's very clear. We can spot the Proterozoic and Phanerozoic difference by the formation of complex fossils. But then we're interested in the origin of life is the last common universal ancestor, right? So the last organism that was or set or group of organisms that was set ancestral to everything that's alive now. And placing that gets to be a little difficult because it could happen anytime between when we see stromatolites, 3.5 billion years ago, right? The sort of the first fossil evidence that there's something alive, which, so probably maybe close to the origin of life or near that. But it could also be as late as the Great Oxygenation Event when everything died off. Okay? And part of that has to do with the way we define life, right? So how do we define life right now? My best definition, the best definition I've heard from other people is the presence of ribosomes, right? Everything we have that has ribosomes is alive, right? And if it doesn't have a ribosome, then it gets a bit iffy. But that's the basic argument there, right? And so, right, but here's the problem with the ribosome. It's a huge complex machine, right? There's RNA involved there. There's several proteins. It's enormous, right? It does a lot of things. There's transfer RNA involved currently, right? So whatever LUCA had these ribosomes, but it also had probably fairly advanced structures as well, right? And so a lot of the origin of life people are thinking about, are there older fossil signatures? Or there's something that we could find that could be older than this, than our last common ancestor. And I'm a biochemist, and so I'm very interested in metabolism first theories. Those sound the best to me. And the one that really convinces me so far is Moran's hypothesis, right? Is that the sort of the oldest signature that we can find for the origin of life is the presence of the TCA cycle. We can look at the TCA cycle. It appears to be something very central. Right? Most of basic metabolism comes out of there. We can get, the formation of lipids, amino acids, all the basic things we need come out of that. All we need to do is put in carbon, and what he's trying to do is show that it's possible to do this cycle probiotic or prebiotically, right? So the formation of these different things, right? So that's what I've been thinking about a lot, is what sort of right? So the idea is that this cycle existed previously or previous to life through some kind of natural geological cycling. And then enzymes arose around that, and life arose around that to take advantage of this cycle. Okay. Again, the cycle as we see it currently is very complex. It might not be the original cycle. Maybe there's something earlier. That's possible, but it's a good running hypothesis. Now, the problem here is, of course, you need complex cofactors, either organic, right? In this case, like the TCA cycle uses, flavins and thiamine, right? All those different things. Or metals, in this case, possibly metals, right? So, and furthermore, you need to get carbon dioxide, or you need to get carbon into the pathway. Otherwise, the cycle doesn't work very well, right? And so the cycle could exist on its own, circling around a couple of times. But if you can't get carbon into it and out of it, it's going to crash and overfill very quickly. And the entrance that everyone focuses on is the acetyl-CoA pathway, right? Which is the bringing carbon dioxide in through acetyl-CoA. But acetyl-CoA is also very complicated as well, right? Now, I don't want to say that I'm 100% like a zealot about metabolism first. It just makes more sense to me. So don't get offended if you're a genomics first person. Okay, I'm not here to start fights. I want to start conversations. Oops, I'm going the wrong direction. Okay. So let's think about where does the idea that the original ancestor was a high temperature organism come from? And it comes from phylogeny, okay? And when the early phylogenies were done, they looked okay, but they had a sort of a statistical issue, whereas that they were having trouble rooting the phylogenies properly, right? The attractor issue, right? Things that were further apart and will change the statistics of the tree. Now, if you go look at the tree, if you root the tree properly and you deal with the proper statistics, it doesn't look so great for the thermophilic organisms, but it looks fantastic for the piezophilic organisms, the pressure-tolerant organisms, right? Which suggests that they're near the bottom of the root. You can see that right here. You can look at the tree on the left, and on the right we have the tree spaced out very quickly. The dark lines are where there's basophilic organisms. You can see some thin lines. They occur in the eukarya, which are of course later. And if you look at the bacteria, the two lines are related to the cyanobacteria and the green non-sulfur bacteria, right? Both of which are photosynthetic organisms. They don't need to be pressure tolerant because they need to be near the surface, right? High pressure systems very rarely, especially at the bottom of the ocean, there's just no light, right? So that's expected, and that's kind of interesting to me. Another thing that I think is interesting with pressure is that favors compressed structures, right? And one of the big puzzles for biochemistry is protein folding. Why do proteins fold, right? And if we can think about pressure as sort of a driver of folding to a certain extent, right? It's a little complex, right? Because proteins, when they unfold, actually reduce the volume, and that's sort of shown here, right? So we have to think about what the protein, when it's folded, will have spaces where there's errors because you're packing spheres together, so you can't pack them perfectly, and they'll also sort of solve and exclude a lot of errors to prevent water from going in spaces. And when you unfold the protein becomes larger, right? The radius becomes larger, but the volume decreases because water can now fill into those sort of occluded spaces a lot better. That's not true with DNA. DNA folding has a negative volume change, right? So that's good. That's favored by pressure. But with proteins, it's a little bit complicated, right? Although having compressed structures is pretty good. And typically up to about 3,000 atmospheres, proteins are usually folded. And so that's sort of stable. That's sort of something that we see. That's a common trait, and so maybe that's something there. It is a little unusual too because protein volume is linear in response to pressure, which is very weird to me. It should be very weird to you because it's volume, right? But essentially what we're looking at is the excluded volume from the protein and this void volume as well. But they respond linearly. Okay. And all the different proteins they've looked at, the void volume, when you look at the void volume, it's linear response. You look at the volume of, and this is all done in crystal structure, so you have to look at the space of unit cell, and that's also reduced linearly as well. Okay. So what else does pressure and temperature, comparison of pressure and temperature get us? We can talk about how pressure affects bond formation, right? How does it affect it? It favors bond formation. Bond formation is also exothermic, which is not favored at high temperatures, right? And that's bad for thermophilic organism life. You have to sort of work against the formation of bonds, right? Bond breaking is endothermic, which is favored at high temperatures, and it's disfavored by pressure, right? So we want to do a little bit of metabolism as well. But proteins and enzymes specifically do also do several things that are also favored by pressure. One is hydrogen bonding. That's very important in protein structure and in enzymatic reactivity. When you form a hydrogen bond, you can actually cover up some of the van der Waals overlap of the atoms, and so their total volume gets reduced. That's good. Like I said, all bond formation is pressure favored. But what's also favored as well is ionization, and that's also favored at high temperature. But ionization, because it changes the way the water can pack around the molecules, can also be favored by pressure and high temperature. Right? And so that's something interesting too, because ionization and formation of charges is a big part of enzymatic catalysis, right? And that's sort of a core mechanism we see conserved there. So the question, can we look at protein ligand binding? And when we do, it turns out to be generally endothermic, again, which is something that would be disfavored, or excuse me. Okay, I'm sorry, I have read it. Okay, generally endothermic, that's binding, but we're looking at mechanisms, and it's not 100% exactly sure what's going on with the mechanisms. And the final thing I want to mention as well is that pressure tends to enhance enzyme reaction rates, right? That's a well-known factor. It enhances the enzyme reaction rate until you start to unfold the protein. Same thing with temperature, right? It's fairly linear. And one of the reasons it may do this is because enzymes themselves are evolved to bind the transition state as tightly as possible. And how do you bind state? When you have a tight binding, what you're doing is reducing the bond length between the atoms. and thus reducing the total volume, which is something, because you're reducing the volume, that's favored by pressure. Right? We can think of enzymes simply as focusing pressure on the formation of the transition state, which I think is kind of interesting as well. So, okay, I'm afraid, I'm worried I might be running a little long, so I'm just going to cut this short. Just go to conclusions really quickly. So what I want to mention is that pressure tolerance is common in modern organisms that are found in the DeLuca. There's some other several interesting mechanisms, several relationships between pressure and the way biochemistry happens that I think are intriguing. So I'd like to remind you that, it doesn't have to be just temperature as the driving force for the origin of life, but we can think about pressure itself as the driving force. Okay? And so thank you for your attention, and